So I think basically the last thing we did was draw the contracted sarcomere, right? Yeah. Oh, let me get my water and be ready. All righty. So I think for your reference, maybe I'll just put a sarcomere up here so we can kind of remember what we're talking about. What's in the middle? Yeah, so you can think M stands for middle. And then what are these two on the ends called? So you could call them Z-disc or you could call them Z-lines. I don't really care. And then where does the myosin start? The M line or the Z line? M line, right? All right, so we'll do our two myosins. And remember, it only goes halfway or 3/4, something like that. So green is our myosin. And do they call that the thick filament or the thin filament? Right. Maybe I'll learn something on Tuesday. Who knows? All right. And then we've got actin. So blue is actin. And then where does titin go? So just from Z to M, and you could put it wherever. You could put it here, put one here, put one up there. This is titin as well. So of course, as you saw last time, when you contract, these myosins, they have little heads on them that pull on the actin. That's what we're mostly going to do today, is talk about how that kind of happens. But from last time, remember these Z lines, they both came closer to the M line when it contracted. So there was more overlap between the actin and the myosin when it contracted, right? So the next part, actually we did do these questions, didn't we? Which gets narrower? And we did this one too? Right, so we're actually here. Let me turn off all the lights. And for anyone coming in late, we are going to finish part 1 and then do our quiz for part 2. If you're wondering what, if we missed it or something, you didn't. All right, so as if we didn't zoom in enough yet on how muscles work, I decided to go all the way. So we're going to go into, we're basically zooming into one little spot where the actin touches the myosin. So you're zooming into about as close as you can get. And there's actually the lab I worked in at CU. Some of the people in our lab were studying myosin. and they could actually put myosin on a slide and then put a piece of actin on it and watch the actin get moved around. And, like, measure how fast it moved to see what was wrong with the myosin and stuff. So you can actually watch this with a fluorescent microscope, which is kind of cool. So how it works, I guess we could start up here on number 1. In red here is the myosin filament. And out of the filament itself, each of those molecules has 2 heads that stick out. And that's the part that's actually going to grab the actin. That's in red up here. Those are the myosin heads. Actin's in yellow, along with some other molecules that we'll talk about later. Obviously, does it take energy to move things? It does. And what does the cell use for energy? It uses ATP. So ATP is involved in this one. I think it actually makes most sense to start kind of between 4 and 1. So myosin grabs ATP. Then it's going to break down ATP and take the energy from it and then move. So the head moves. And you can see that here. It moved from a little straight up to a little more to the right. Now, is it already pulling on actin or anything? It's not. So it's basically like cocking a spring or something, or you're like storing energy in that movement. So you take ATP, you eat it, and then you turn like this. And then you wait till you grab onto actin. So then step 2 is to grab the actin there. That's step 2, you grab onto it. Step 3 is you use your stored energy to pull it. And then step 4 is bind ATP and let go. So step 1, you eat, use your ATP, turn, grab on, pull, grab ATP, let go. That's kind of the steps. So we've got our little random piece of trash that I found around to use. Who wants to come be a myosin? Use like 4 or 5. All right, there's 1. Who else? Thanks. All right, how about one more? I got Jordan. And then we also need a Z-disc. Who wants to be a Z-disc? All right, Pedro will be our Z-disc. All right, so let's put Pedro over here. So you're there. Now, how do you think our myosin should line up? Like this way, this way. Oh, maybe I should mention. So that's one Z-disc. The other Z-disc's over there. All right. So here's your Z-disc. So, and this, what do you think this tubing is? Well, it could be titin or actin, but we'll call it actin. And you can just imagine M line would be way over at my desk over here. So if this is our M line in the middle, where should our myosins be then? They start at the M line, right? So I should go over that way a little bit. All right, and so we're starting here with our actins. So we're starting with our Z line attached to actin, but we're relaxed right now, right? So step 1, can we get your myosin heads ready? All right, so then we'll feed them some ATP, and then what happens? Well, before you grab onto it, you eat your ATP, and what happens to your, maybe go like this. Okay, so we shifted, so that was step 1. Step 2, now you grab onto it. But remember, you were in your shifted state when you grabbed it, right? Yeah. Step 3 is you're going to pull on it. So you just moved your Z-disc over a little bit. And then step 3 is what? Grab ATP and let go. Now, that's fine, but what if we want to keep contracting? Can you all let go at the same time? No. So what happens is maybe one of you is still holding on while the other two grab new ATPs. So let's have, Rosie, you just hold on. Your job is to keep held on, and y'all's job is to pull.
Okay.
All right. So, yeah. So you eat ATP, turn it, grab it, and then you pull it. Now you're going to hold on.
Okay.
And you can eat ATP and let go. And then use it, rotate, and then pull some more. Now, if the nerves stop telling you to contract, what's going to push Pedro's Z line back if we let go? Yeah, so tighten. So you got to imagine that there's like a crazy, actually the spring doesn't quite work. What we really need is a spring. So you can imagine a spring getting tighter to me, and then when the myosin lets go, it pushes Pedro back along with the actin, right? It's going to get pulled all the way back. So that's your 4 steps of contraction. So a round of applause for our volunteers, our myosin. Good job, good job, good job. Good job, Z-line. Let's look at that later. All right. Well, actually, can I find a better one? Oops. All right. So let's try. Before we do that demonstration, let's just take a quick look at this. All right, so here's our contraction cycle.
The release of calcium ions into the sarcoplasm, initiating a contraction cycle. A single contraction cycle consists of 4 steps. First, ATP, attached to the myosin crossbridge, must be hydrolyzed to provide the energy needed to move the myosin protein.
Hydrolyzed means broken down, and you take energy from it. So if you saw that myosin head, which way did it move? Take a look at it one more time. See which way it moved.
A muscle action potential triggers the release of calcium ions into the sarcoplasm, initiating a contraction cycle. A single contraction cycle consists of 4 steps. First, ATP, attached to the myosin crossbridge, must be hydrolyzed to provide the energy needed to move the myosin protein. Second, actin and myosin filaments temporarily bind together. Third, the release of the phosphate molecule triggers the power stroke. The myosin filament rotates, releasing the ATP. The force of the rotation slides the actin filament toward the center of the sarcomere. Fourth, the myosin head and actin are detached when another ATP molecule binds to the crossbridge. This ends one contraction cycle.
So that's your 4 steps. So I think we should all stand up and do it then. Just to put it in your head so we can all stand out this way. All right, so let's see. Everybody face backwards. And let's say our M line is way over on the left. The Z disc is way over on the right. And we'll just pretend that actin's going all the way at the moment. So step 1, you got your myosin heads. They're up. They're unexcited. So they're going to bind to ATP, or they have ATP already, but they're going to break ATP down, and then what happens? So you're going to shift towards the Z disc. That's step 1. Step 2, grab your myosin, or actin, imaginary actin, got it on. Step 3 is to pull it. Step 4, half of us are going to grab an ATP, and that makes us let go. Because we can't hold on to actin while we're looking for ATP. So you're going to get your ATP, turn. So use ATP to rotate your myosin heads, grab it. Now the people that were holding on before, they can let go. And now we can pull it. And so we just moved imaginary wall closer to the left. So that's your 4 steps there. I expect y'all to be doing that at home to practice. Yeah, if you'd like, on your test, if you want to come up here and act out sarcomeres for me instead. Maybe. All right, so let's do some questions and see if you got it. So for all these, for all these questions, it's going to have the same answer choices. So put this. A is stage 1, B is stage 2, C is stage 3, D is going to be stage 4 there. Or E is none, I guess. All right, so first question. Which of those stages the myosin heads bind ATP, causing the cross bridges to detach from the actin? Seven, 9, 18. Answer choices are up here in case you weren't sure. All right, so almost everybody said step 4 up, which is correct. So right there, you can actually see it letting go. Right. So during the contraction cycle, ATP attaches to what? What does ATP attach to? All right, so I want you to talk to your group there. It looks like you're tied between A and C. In case you weren't sure what the question was, ATP attaches to what? All right, final vote. All right, so nearly everybody changed to C. C is the correct answer. So myosin's the one that grabs ATP and uses it, breaks it down. Like when we were standing up, we were the myosin, we ate the ATP. Actin's just the rope. It just gets pulled on. All right, so guess I can just hold on. This is silly. Let's just make this question make more sense. All right. So during the contraction cycle, ATP is hydrolyzed, which process is fueled by the energy released. All right, so maybe try with your group there. Where'd she go? All right, final votes. So the answer is D there. Got up to 60% from 40. And I think what people want, I don't know, it's hard to think of it as all these separate steps. That is how it happens. So when you break down your ATP, remember all you do is this. You're not grabbing on anything, you're not pulling on anything, you're just storing the energy basically. Why is it that way? Probably because that's the most efficient way that evolution could figure out. Is to go ahead and do this first and then just use that stored energy after you grab it to pull. And actually, similar, myosin's all conserved. So you see different kinds of myosin, but they all work the same way, basically in all the cells, different kinds of cells. All right, so I think I put the rest of the stuff in part 2, unless I don't remember. Make sure. I guess not. We'll go back to that if we have time, but otherwise you can just read over those few slides for like what happens during exercise. There's just like a slide for each one. Weird. All right. So questions for part 2 study guide before we take our quiz. Yes, so how do muscle fibers regenerate, which was objective C. What is that?
So it's when like
So yeah, you are right. But to keep it a little simpler, there's two main ways your muscles can repair themselves after they're damaged. So the main way is that the muscle cells that are there get bigger. And then the second thing, and maybe a little less common, is stem cells that are in the muscle divide and make new muscle cells. You mean like really cut into it? And so for something more severe like that, you'd probably have more of the new cells. And then the other ones would get bigger too. But for something more mild, like you're just lifting weights and, feel sore, you're probably just mostly just making those cells bigger rather than forming new ones. Sure. So function of each type. Skeletal is just basically for moving the body, stabilizing body positions. Exactly what you'd think of, probably. What's cardiac muscle's job? So pump blood in the heart. And then smooth muscle, it's got all kinds of different functions. So change the size of your blood vessels, move things around your body, like your GI tract, your stomach, help with breathing. Maybe leave that last one off. Let's not do that one. So that just gets confusing. So yeah, move things around your body, and then change the size of your blood vessels would be two good ones to know for smooth muscle. And actually, while everybody has their study guide out, you can cross out the last one on part 2, where it says explain the 4 phases of a twitch contraction. You just take that one off. Alrighty, let's take the quiz. right, so pass those on up. You're done. All right, so which of these is the muscle cell membrane? Sarcolemma, T-tubule, sarcoplasmic reticulum, or myoglobin? This is the sarcolemma, exactly. Which of these constricts blood vessels? Smooth muscle. Which of these is muscle cell smooth ER? It's the sarcoplasmic reticulum. What does the sarcoplasmic reticulum store in muscle cells? It is calcium, mostly, definitely. What is a motor unit? Sarcomere, group of muscle cells activated by one neuron, group of myosin heads. It's a group of muscle cells activated by one neuron. So within, like a bicep, there's lots of motor units because there's lots of different neurons coming in there. So anyways, what actually does the work of contraction? It is the myosin, right? Right. Let's do the old pile here. All right. So you kind of have a good idea of how these sarcomeres work. But what's left is to talk about how that sarcomere knows when to contract and when to relax. So first off, what tells your muscles to contract? So brain, nerves. Is there any time where your brain's not involved and your muscles contract? Oh, cardiac and smooth, that's true. What about for skeletal muscle? Is there any time where the skeletal muscle contracts without the brain telling it to? A what? Maybe a toxin. Could be some kind of disorder. What about reflexes? So if you put your hand on something hot and you pull away, that's actually a signal sent to the spinal cord and back out. Never gets to the brain. So when we say skeletal muscle is voluntary, that's mostly true, but not completely, even though I know it says it in the book, and most people would say that, but reflexes aren't voluntary. You can't control them, right? So, but that's still a nerve telling the muscle to contract, right? So a nerve coming back from the spinal cord and then sending a signal to the muscle. So most of part 2 is seeing how that works. How does a nerve get this sarcomere to contract? So we call that the events at the neuromuscular junction, which just means where the neuron meets the muscle. All right. So there's a few steps involved. I think this will make more sense. Let's go this one first. So here's all your skeletal muscle fibers, which are cells. And then this lighter colored thing is the neuron coming in. So you've got the axon. And then in multiple places, that one neuron has a little extension, a little cell membrane outgrowth that touches the muscle. or comes really close to it. So you could see one here, you could see one up there. So that way it can tell that muscle cell in multiple places to contract, and then tell the cells around it to contract too. So we're going to be zooming in on like one little spot there. So what happens there? Let's do the words first, then we'll do the picture. Has anyone heard of acetylcholine before? Does that sound at all familiar? So it is a neurotransmitter, which means it's a molecule released from neurons that goes over to another cell and tells that cell to do something. And the way nerves tell muscles to do something is the signal gets to the end of the nerve releases acetylcholine, it goes over to the muscle cell surface, not inside, just on the surface, binds to a receptor. That receptor then opens up and lets sodium ions in. What charge do sodium ions have? They are positive. What's the inside of a muscle cell usually, do you think? Negative or positive? Any guesses? It is usually negative. So the muscle cell is usually negative inside. So when this receptor's told to open, lets all these sodium ions in, what happens to the charge inside that cell now? It's more positive. And that's going to be an important signal in a moment. So we're going to continue on from step 3. But also important is that acetylcholine, that signaling molecule, it's degraded very rapidly, which is good because how long do you want your muscle to contract after you tell it to? Not very long, just long enough to get it done. If you want it to keep contracting, you keep telling it to. Otherwise, you want it to relax. So the way you maintain that is very rapidly get rid of this acetylcholine that you released. So yeah, you could have too much acetylcholine being produced. Also, there's occasions where the myosin doesn't let go of the actin. So for example, do you remember what makes the myosin let go? Grabbing an ATP. What happens if you run out of ATP? Right, you get stuck holding on to it, contracted. And that's actually where rigor mortis comes from. People are like contracted and stiff when they're dead. That's because of this. The myosin, there's no more ATP to grab onto, so it just gets stuck, attached, and everything gets contracted. It's kind of interesting. All right. So this is exactly what we're talking about, just in word or in pictures. So up here in yellow is the nerve cell. Down here is the muscle cell. So number 1, acetylcholine is released. That's the red thing there. So that travels across this little space, and it binds to these receptors in green on the muscle cell. That's step 2. Step 3, those open. So whenever that acetylcholine binds the receptor, it makes it open. And so you can see that open there, and it's a channel that lets sodium ions in. And then step 4, you're breaking down the acetylcholine so that you're not sending any more signals. Oh, and getting back to that cramp thing for a second. How do you fix a cramp? Well, do I? Yeah. Have you seen them fix it at like sporting events or something? Rub it a little bit. What else do they do? they stretch out like crazy, right? And that probably hurts. Has anyone had a cramp fixed like that? It doesn't feel very good. So what's happening when you're stretching it is they're actually forcibly breaking, pulling that actin off the myosin, basically, which probably is going to damage a few of those filaments and cause some issues there. But that is why it works, because you actually just break those. All right, so anyways. One word we didn't talk a lot about is that action potential word. So action potential is the cell gets more positive inside. That's what an action potential is. Cell gets more positively charged. So which of those is directly responsible for transmitting action potential from a neuron to a skeletal muscle fiber? All right, so I'll let you talk to your group for a moment. We'll let you vote again. All right, final votes. All right, so it is either acetylcholine. It's the neurotransmitter. Oh yeah, somebody voted for F. Oh well. All right, so we've got our action potential, but we haven't gotten to the point of contracting yet. So that's what excitation-contraction coupling is. So if you remember, that signal led to a lot of sodium ions coming in to the cell. And what that does is it opens up calcium channels on the smooth ER of the muscle cell, which we call the sarcoplasmic reticulum. So it opens those channels, and all this calcium comes out of the SR and into the cytoplasm. Normally, the cytoplasm has almost no calcium at all. And so open all these channels, you get a very rapid increase in calcium. Why is this thing covered up here? All right. So release calcium. And then there's a few things we haven't really talked about. So the way you prevent contraction from happening without signals is actin actually has something covering it up. So it's got stuff on it that physically block myosin from grabbing it. And that's actually called troponin. It's two molecules, troponin and tropomyosin, together. So they're covering up the actin so myosin can't grab it. And calcium comes out, goes over to the troponin, tropomyosin, and makes it move out of the way.
I know, like in cardiac patients,
Mhm.
they always test for the troponin levels, like to see if they've had a heart happening or any heart damage.
Yeah, it's like released during with heart damage.
That's released or like if something goes wrong, there's too much of that is released, and is that why the heart like stops beating?
I wouldn't worry about the heart thing right now, but yeah. So calcium comes in and it binds to these and it makes them move out of the way. So like move to the top so now myosin can grab it. So that's step 4, 3, 4, and 5. So calcium binds to this molecule troponin, makes it move, and now myosin's able to grab actin and pull it. Titin's separate.
So then where's the, how do you say,
Troponin.
where's that come from then?
What do you mean, where does it come from?
Like, positive form, like why is it there? Well, I mean,
Yeah.
why it's there, but where's it come from?
So basically it's always sitting on actin. It's not really attached to M or Z. It's just a small molecule that sits on actin and covers it up. And we've got a few movies in a little bit too that might help it make more sense. So I know this is a little complicated. So you can kind of see it on here. You've got your relaxed state. So if you see this blue and brown kind of thing wrapping around the yellow actin, it's covering up the binding sites. So do you see these little black circles on the yellow? Like if it's a black-eyed pea or something? So they're all touching that brown troponin, right? They can't really see anything else. So if myosin tried to grab it, it has to grab on those little black dots. So it can't grab them right now because they're facing the wrong way. So when calcium comes in, it's indicated in purple here, it binds to the troponin, tropomyosin, and it makes it twist a little bit. And so now you see how these black little eyes are sticking out? So now myosin has something to grab onto. So relaxed, and then calcium comes in, moves us out of the way. Now myosin can grab it, and we can do our whole contraction cycle that we were just talking about. Those 4, grab ATP, use it, grab onto it, pull. And so long as calcium's around, actin's exposed, and you can keep pulling on it.
So is that not only happen in skeletal muscle system, where there
Everything except those first few steps of the nerves. This happens in all of the types of muscle tissue. The calcium's released and troponin moves out of the way. 4 steps of contraction. It's just that first little signal that gets everything started is somewhat different. And when we do, if you take 202 and we do the heart, we'll talk more about how that's signaled. But we'll just do skeletal muscle for now. Yes? Up here, it's just the structure of the sarcoplasmic reticulum. It's just kind of saying where it is. It's not really important for us. All right, let's see which way to do first. All right, so I think what we'll do, I've got 2 paragraphs to correct for you, but first I've got like a 4-minute movie that goes over all this. It's actually it's got really good visuals, a lot better than the ones that came with the book. So what we'll do is we'll watch the movie once, let you read over the paragraphs, try to find some mistakes.
Muscles every day to do it.
Then we'll watch it again. let you correct the mistakes with your group after that. Because I know there's, I mean, how many steps did we just put this into? Like 12? It's a lot of steps. All right, so let's get on to our skeletal muscle. All right, get this out of the way.
Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contraction.
What made that actin go back when it relaxed? So it's the tighten that's not shown on there anywhere, but you know, when you pulled that Z line, you're like compressing that tighten spring. And whenever you let go, it just pushes it back. So, anyways.
Are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin.
So those little yellow dots, those are the myosin binding sites. You can see they were covered up and now they're exposed.
This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from the neuron and bind to receptors, which depolarizes the membrane of the muscle fiber.
What's another way to say that? What's depolarizing? You know? What happened when those neurotransmitters bound those receptors? It what? Not quite. It made them open, and it let something into the cell. Not quite. Calcium's later. ACH was the signal that went and bound the receptor. It is sodium. So acetylcholine came and bound these receptors, opened them up, and it let sodium in. So when they say depolarization, that's just a fancy word for letting sodium ions in. That's what they're saying. So.
The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
All right, so we'll take a few, 5 minutes or so. We've got 2 paragraphs to look over. See if you can find any mistakes. If there's something you think maybe that doesn't sound quite right, maybe like circle it. And when you watch the movie again, maybe you could like look for that little part. Does that kind of make sense? So, do 2 tries. Here we go. Don't look so excited. Got some extras, I bet.
We're supposed to do this on our own, right?
Yeah. So try this one on your own first for 5 minutes. See if you can find anything in there. right, you ready for the movie again? See if you see anything you didn't notice before. Right. So I got it started at about a minute here.
Called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibrils. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M-line. The thin filaments are composed of the protein actin, which are anchored to the Z-lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z-lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M-line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which polarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z-lines draw closer to the M-line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten.
All right, so work with your group now, and I'll tell you there's 6 mistakes in the first paragraph and 5 in the second. So maybe all your brains together, you get most of those. 6 in the first one. 5 in the second.
No, it's calcium.
Just the size. All right, so let's go over that and see how many you got. If you got most of them, you're doing pretty good. So these are quite the paragraphs. This is the first time we've done these paragraph corrections. They're one of my favorite things for processes. So you'll see more in the future. Oops, sorry. I gotta find a place to store that. All right, so first sentence, neuromuscular junction is in your brainstem, is that right? How did you fix it? So something about it's in the muscles, it's where nerves meet muscles, anything like that would be fine. What about the second sentence? Neurotransmitters released from the neuron axon terminal. Anything wrong with that? Nope. Release of neurotransmitter is triggered by signals from the brain. I'd say that's correct. That's something that wasn't maybe mentioned in there, but the reason that neurons releasing those transmitters is because the brain sent a signal down all the way through chain of neurons to get there. This next sentence is a real mess. Let's see. Neurotransmitter then moves into the muscle cell. Is that right? So you could just change it to goes to. So it goes to the muscle cell and it binds to acetylcholine receptors in the cytoplasm. Yeah, it's not the cytoplasm, it's cell membrane. Or you could have said sarcolemma if you wanted to describe it that way. So cell membrane or sarcolemma. This triggers sodium channels to open in the cell membrane. Is that right? The way it's written is correct. So where people are always getting confused is it is sodium channels first. So don't skip ahead to our calcium. Sodium's the first signal. So this causes sodium channels to open, so it's written correctly. As a result, the voltage inside of the cell becomes more negative. It's more positive. It's negative to positive. Neurotransmitter is very slowly degraded. It's quickly degraded. So signals sent to your muscles are quite long-lasting. You could say not, or they're short-lasting. Somehow make it be not long-lasting. So that was our 6. Did anybody get all 6? I didn't think so. I put a lot of tricky ones in there. All right, so number 2. First sentence, the voltage change generated at the junction starts a muscle contraction. That's right. This voltage changes, causing potassium channels in the SR to open up. That's our calcium channels right there. This causes calcium ions to flood into the cytoplasm. That's right. Calcium ions bind to myosin. Yeah, so you could have said troponin, tropomyosin. I just think of them as one molecule together. And so you also need to cross out thin filament, right? Oh, I'm sorry. I got on the wrong spot. No. Calcium ions bind to troponin. The binding causes troponin to change shape and exposes myosin binding sites on actin. That one's good. Myosin then uses its heads and ATP to pull the myosin. Should be actin. Myosin doesn't pull on myosin. Myosin pulls on actin. Contraction continues until neurotransmitter stops being released. That's true. After the signal from the junction disappears, the calcium is pumped into the mitochondria. Should be sarcoplasmic reticulum again. Or SR, because we abbreviate it. All right, and then the lack of calcium causes troponin to change shape and cover up myosin binding sites on the thick filament. Should be the thin filament. Exactly. All right, so one last slide here. Now that you know how muscles contract, obviously if you're like doing curling barbells or something, at some point you just can't do anymore, right? No matter how much you're telling your arm to lift it up, it just can't. Even though it could for a while, for 20 reps or 30 reps. So thinking about contraction, why is this? You're not releasing calcium ions anymore from the SR for some reason. Maybe you're just out of ATP in the muscle cell, because no ATP means you can't do the whole contraction cycle. Lactic acid building up, because you go into fermentation when you run out of oxygen, and lactic acid will turn it off somehow. And maybe you ran out of neurotransmitter that neuron just kept sending and signaling, and now it doesn't have anymore. So it's actually kind of interesting. They don't know exactly which of these is the primary reason why your muscles just won't contract anymore. It's kind of hard to study, but
When you're sore, you get tired quicker. I thought you would
yeah.
why does that?
So the soreness is from the lactic acid buildup, and it's also from those fibers do tear somewhat and it causes inflammation, and then they're not going to work as well for a while. Yeah, and then some of the fibers are actually torn a little bit, microscopically torn, not like a torn muscle like you think of as like an injury, but that long muscle fiber, and maybe it's not quite as connected as it used to be. And so it takes it a couple days to reconnect it. So that's your fatigue. So we've got one more chapter till our test. So what do you think you should do over spring break? Study. Which chapter's harder, 9, 10, or 11? 10. So best way to study 10. 10's our first process chapter where we've got step after step after step after step. So probably the best way to study that is to come up with basically, I would say 3 paragraphs for the chapter that you've memorized and you can write without looking at anything. Probably ask you to write one of them because we've got our, neuromuscular junction events. Then we've got how that leads, the excitation contraction coupling is another one, which are the first, that's these two. And then the third one is how the sarcomere contracts. So if you can draw a sarcomere, write those paragraphs, you know this whole chapter. Yeah, because if you don't know how it's put together, you're probably gonna have a hard time remembering how it all moves. So this will be different from how you memorized all the bones, and it's a very different sort of studying. Right.